Morning, everybody. Thank you for joining this session. We have some great speakers for you this morning, and what we're going to do is start out with a short presentation from each one, and then we'll start asking questions. And they'll ask you questions, so like I said, you guys and then, of course, you ask us questions as well, no doubt. But I want to introduce first Dr. Dennis Kyle. He's in the session, a health professor with the University of South Florida. He's a distinguished university health professor in the Department of Global Health at the University in Tampa. He majored in biology at the University of Tennessee at Chattanooga and completed his PhD in zoology at Clemson University. After a postdoctoral position at the University of Georgia, he worked at Walter Reed Army Institute of Research for over 20 years. And then during that time, he led key efforts with the U.S. Office of Drug and Vaccine Development Programs, eventually serving as Deputy Director of the Division of Experimental Therapeutics. Wow. He also served at the Armed Forces Research Institute of Medical Sciences in Bangkok, Thailand in 1991 through 94, and was a senior scientist at the Australian Army Malaria Institute from 2002 to 4. I was speaking with him last night at dinner, and it's just amazing, all that you've done. We want to hear more. Okay, so his research interests include elucidation of mechanisms of anti-malarial drug resistance and discovery of new anti-parasitic drugs for diverse disease, including malaria, visceral leishmaniasis, and primary meningoencephalitis. He's a fellow of the American Society for Tropical Medicine and Hygiene and the American Association for the Advancement of Science. And his lab is supported by research funding from the National Institutes of Health, the Bill and Melinda Gates Foundation, and Medicine's Malaria Venture. So Dr. Sloan.
Thank you very much, Donna. So I just put together a few slides. I didn't want to get too technical, so if I say something that you don't understand, just stop me. What I want to do is try to give you a flavor for what we can do and what we're trying to do in the lab to discover new drugs for these pathogens, including Naegleria phallonoide. No disclosure. So one of the problems is we've heard, even though we've had this great case this year of Sebastian, another survivor, I still think that we need newer drugs that are very fast acting. I won't go into all the technical detail today, but one of the things we've done in the lab is really focus on trying to identify a drug that acts very quickly. And most of the techniques that were used before we started our project required exposure to the drug for more than 72 hours to see activity. In fact, in our hands, it takes 72 hours for miltefosine to actually show activity. With a really rapidly fatal disease like this, we need something that inhibits the amoeba very quickly. So that's what we're focusing on, is trying to find something that works faster. I think the other major unmet medical need, you heard a little bit about from Ibne this morning, we do have diagnostic techniques, but we don't have something that's really sensitive, specific, and fast that's right at the bedside. And I think those are the two key things that we would like, at least I think we should be focusing on. Why is there so little progress? There's so few people working on this. Big drug companies are the ones that mostly make new drugs, and none of them are working on these diseases. And even if you look in academia, university research, unlike malaria, the disease you heard that I work on a lot, there's thousands of labs around the world that are trying to discover new drugs for malaria. There are a handful that are actually doing that for these diseases. So the scale of what's going on is very small. And you can't do anything in a vacuum without any funding at all. So trying to do research on these pathogens is limited because of that funding. So I won't go into great detail, but if you look at how new medicines were discovered, there's a series of different kinds of techniques, and it's too small to see on the screen, so I'll just go to the next slide and focus that. If you look at just this review that came out a few years ago, 259 different drugs that were approved by the FDA between 1999 and 2008, 75 of those were new. That means they were completely new to medicine. Nobody knew about them before. They had a MMOA, a molecular mechanism of action, which was different. 50 of those were small molecules like drugs, and some of those were biologics, more like antibodies that would work. And you don't have to know all the details of this, but phenotypic screening was successful in many of those. And basically what this means is you don't have to know anything about the biology of that disease. You just have to be able to put the cell with the drug and you find a result. So you don't have to know which proteins, which targets in the organism you should be going after. And that's a problem with us because we don't know those in the amoeba. So phenotypic screening, even though it sounds kind of haphazard, you're just screening lots of molecules to try to find an active, actually gives many of these new drugs. So some of the advantages are you don't have to know anything about how they work. You can find them just because they, in our case, they kill the amoeba. We don't have to know exactly how they work, even though they do. And there are some limitations. Many times the throughput, how many compounds you can test, is actually smaller than a target-based screen where you have an isolated protein. But not really, that's not going to be a limitation for what we're doing with amoeba right now. Technical expertise may not exist. Many places are not going to want to grow Naegleria fowleri in the lab because of the pathogenic potential, so only certain centers are going to want to do this. And we still may require to understand it before we can go forward. So why do we do this sort of approach with Naegleria and acanthamoeba amoeba? And I'm going to sort of throw those in the same hat today. We are working on both at the same time in our lab. We're hoping that we can come up with something that will kill both of those in the cerebral, in the CNS disease. First of all, Francine will probably tell you more about this. It's a very complex biology of these organisms. We still don't know very much about them. So trying to identify what we call a druggable target, that means like a certain kind of protein or a certain biochemical pathway that we know if we hit it with a drug, it's going to kill the amoeba. We don't know those in this organism. So we can't really go after the typical target-based screen. At the time we started, there were really no high throughput assays. That means, can you put a lot of compounds on the amoeba and test them very quickly? In the old days, you could only do handfuls of compounds. Chris is in the room. He's just tested over 30,000 extracts in a few months by doing the sort of methods that we've come up with that he and Beatrice basically came up with in the lab. And then a lot of expertise is not available in industry for these. So this is a slide that sort of tells you what is the approach that we're doing now in 3 different steps. The first one is, this is the same for any kind of drug discovery. So it's not unique for amoeba. It could be cancer, it could be other infectious diseases, any metabolic disease. All of them require that you find something that works, and then you optimize the properties so that you can take it by mouth or you can give it by injection and it actually gets to the target and has its activity. There's a big difference between a test tube where it shows positive and actually a drug that you can give to a person. So we first try to find the test tube actives and then make it into the drug. We call that process lead optimization. And I'll tell you one story that we're doing now where we're doing that for these amoeba. The second one is you screen large libraries of either small molecules or natural products, and I'll give you more reasons why we do that in a few minutes. And the idea here is that you screen those, you find those hits, and then you do the optimization and make a new drug. So this is where you already have something you know is active. Here's where you're trying to find something new. And then the third one is called repurposing now. It used to be called lots of other things. But basically, you're looking for compounds that already have activity against other diseases. And in our case, we were looking at over 1,000 compounds that had either been FDA approved for something else, have been in clinical trials for something else, and seeing if they have activity. And I think everybody knows miltefosine in this room by now. Miltefosine is an example of that. It was actually an anti-cancer drug. It was found by a friend of mine, Simon Croft, in the London School, that had activity against visceral leishmaniasis. That's what it was developed for. And then once it was developed and approved, now we find out that it can be repurposed for amoeba. So we're trying to do that as well. So I'll give you a brief example of each one of those. One of the first series we started on, and I didn't show much chemistry because I didn't figure this audience would be really excited about a bunch of hydroxy chicken wires, I call it. But basically, the idea here is that we've identified some compounds, and we know about these. Pentamidine, propamidine, which is used for keratitis. These series of compounds have activity against amoeba. The problem is They're not so specific against the amoeba, and they have some toxicity. They don't really get to the blood-brain barrier. But one of the chemists that I know very well at Georgia State had identified some compounds here. And the cool thing about these, you can just ignore all the numbers except look at the cures here. This is a different disease, but it's also a central nervous system disease, another parasite. And this was proof that these compounds actually made it across the blood-brain barrier. So our hypothesis was we can take this series, we can identify some that might cross the blood-brain barrier, get to the amoeba, and identify something that's working. And put that down, I'll keep pushing the button. Again, more chemistry. What we did was got the chemists, we screened over, not all 2,000, we had a library of these analogs. We tested them, we found the 2 that were the best, the 2 on the top. And now what we're doing in the lab is trying to optimize them. And this is a graph that Beatrice put together from some of the data. And these blue and orange lines, what that shows is the orange line, orange dots are activity against the amoeba. So this is weakly active, this is very, very potent. So you can see what we're doing is we're making more and more potent compounds, but notice how it's separating from the blue dots, because we want something that kills the amoeba but not harms mammalian cells, people's cells. So this is the process of lead optimization. The cool thing for us, and I won't go into all the details, we now have a whole series of compounds which are active at very low nanomolar concentrations. This is over 2,000 times more effective in our hands than miltefosine or anything else. So it's really exciting. We have these very active compounds. Now can we turn those into drugs? That's our ongoing battle that we're working on. So I said that's the lead optimization process. Where do you find these other compounds if you don't know, if these don't work, where do we find the other hits? So we started with a malaria box, and I won't go into great detail, but there's a series of bioactive compounds. We just screened them, and what we found was that out of this 400 compounds, which were already known to have some activities against other parasites, here are 5 different compounds that came out. And again, these numbers represent very potent activity. That's 1.5 micromolar and 190 nanomolar. So these are very, very active compounds that we found, again, by just screening a library. Once we have those, now we start working with a chemist to try to turn those into new drugs. So what we're hoping to do, and this is always a problem in drug discovery, if you pick one type of chemical and it fails, then you have to go all the way back to ground zero. So what we're trying to do is take our best ones forward. Drug discovery is basically a process of failure. Most things fail, so you try to push it as fast forward as you can to fail as quick as you can, and then you have these backups. And so that's what we're doing. We're filling the pipeline in our lab to have a lot of different compounds that will have activity, and then we can progress those. And there is a trick to that, because if you have 200 compounds that are active, they're all different. How do you pick? Well, that's the kind of thing that we're doing. We're coming up with ways to test that. We also tested from another library I won't talk about. The other thing that you would be interested in, I think, is the screen from St. Jude. St. Jude is a pediatric specialty hospital in Memphis, Tennessee. A good friend of mine there, Kip Guy, has been running the chemical biology or drug discovery program for cancer and infectious diseases. And we've been working with him to test over 1,300 of these bioactive FDA-approved compounds. We're kind of excited because we're hitting the kind of things that might lead us towards coming up with something like the next miltefosine that's a repurposed drug. And one example you heard this morning, azithromycin. We hit that very well. In fact, azithromycin is much more active than what I remember from reading in the literature in our hands. It works very, very quickly. It's very potent. It may take a little bit of time to work, but it's those sort of confirmation that we're trying to work through. So right now we have 4 compounds that work against both Acanthamoeba and Naegleria that we're trying to work through. And then the last one I want to give you an example is kind of exciting for a different reason, and that's these small molecules from natural products. So if you look at the history of trying to find a new drug for any sort of disease, many of those came from natural products. Now, a natural product is some microbe is trying to prevent itself from being eaten by another microbe,
And so these metabolites that they produce actually are active to either taste bad or make the other organism not want to kill them. And what we're harvesting is this natural ability of these organisms to make these products. And then we find those, and that's how you find all sorts of drugs like Taxol and other compounds all came from these natural products. So what I did was work with two friends of mine that I've been working with on a malaria project. Cedric Pierce, who's at a small company in North Carolina called Mycosynthetics. He has, I think, the world's largest library of these slow-growing fungi that he's come up with several new leads for drugs. And Bill Baker, who's an organic chemist, a natural product chemist, who isolates exactly what is it that's in that mixture that causes the organism to be killed. And he also has this unique collection of microbes from Antarctica. So it would be really cool to have an Antarctic microbe that kills a brain-eating amoeba. So very briefly, as I mentioned earlier, we used our new techniques for screening. We've screened over 30,000 extracts. From those, we found a number of actives. So now we're hoping that NIH will allow us to continue, because we actually did twice as much as we promised. We said we would do this for Naegleria fowleri in less than 6 months. I think we did it all in about 4 months. And we also did the same thing for Acanthamoeba at the same time, which we didn't even promise them. So we have some really interesting leads there to go forward. And now we're hoping that we'll get the funds from NIH to be able to now identify what is active in those. And those could be the next generation of our drugs for the organism. Here's something that Chris put together from this. You can see 25,794 compounds were inactive. And what we're interested in is this little sliver of red there, where the most active ones. And you can see even at low concentrations, we still have 197. That's a lot of new things that we can start to work on. The last point I want to make, and this is something that we were hoping would work. Many people, as I mentioned, don't want to take Naegleria fowleri in their laboratory and do this sort of work because of the pathogenic potential. So we were hoping that we could use something related but non-pathogenic, and that's Naegleria lobenensis. It's supposedly the most closely related genetically to Naegleria fowleri that we know. It grows at 37 degrees, et cetera, but it's not pathogenic. So we've done this at least 3 times with different libraries, and what this shows you is that if you screen with a pathogen, we found 34 hits. If you screen with lobenensis, you get 19. What we've found repeatedly with different libraries is that if we don't screen with a pathogen, we're going to miss things. We may find the same hits, like azithromycin, but we may miss a lot of hits that we would get. So this shows you that you can't avoid working with the real bug if you want to do this sort of discovery. So what we really want to see is something that, again, works on Naegleria fowleri and Acanthamoeba. That's our goal, but if we can find a new drug for Naegleria and find a different one for Acanthamoeba, we'll still be happy. So the end goals of this, I think this is an important thing, to maybe a jump off point for the discussion later, is what happens if we're successful, okay? Well, first of all, how much industry is going to be interested in developing this is probably pretty limited. So biotech or other small pharma might be interested in taking it forward. We are working with the National Institutes of Allergy and Immunology, Infectious Diseases that have some capabilities for drug development. So some of the things that we couldn't do on our own, we can go to them and seek resources. We write a white paper. If they approve it, then we get them to do some of that work. We could go to something like the NIH Rare and Orphan Disease Program. Or ideally, I think what we should be shooting for is something like a compassionate use IND, where you get safety data and show that it's probably safe to use. All the laboratory and animal studies suggest that it's efficacious, and then define how it's going to be used and identify somebody to actually hold the IND. CDC's held the INDs before. Other groups could hold that. But that's probably where we're working in that sort of range, and I'd be glad to have a discussion about what people think about that. And I just want to close by thanking my team, which is all here. Beatrice is right there. Raise your hand. Don't be shy. She's a PhD student focusing exclusively on Naegleria. She's doing some of these quick assays, killing or not killing the drugs. Chris did his PhD in Acanthamoeba at the University of West Scotland. And so he brought expertise in Acanthamoeba, but he's also doing a lot of the screening I just mentioned on Naegleria. Kadi is a master's student, and she's developing a thesis on this. She's helped a lot with some of the screening. And Caitlin's an undergrad who's done a lot of screening that I just told you about. And then all the groups, the chemists at Georgia State who are making the new compounds that we are really excited about, the natural products, Kip Guy, and then of course funding, which is a limitation. So, but we're very happy. NIH has been supportive of what we're doing. So that was sort of an introduction, and I think you want to introduce the next one? Okay.
So, Dr. Lee, so while you're bringing yours up, I'll go ahead and talk about this.
See if you can find it. That's me.
Dr. Cabral is, as I mentioned, professor researcher with the Virginia Commonwealth University School of Medicine, and she's very humble. Okay, she's very humble, but I'm going to talk about her
No,
anyway.
you don't have to.
Yes, she's amazing. Actually, little known fact, I mean, in 2007, when we had the 3 cases in Orange County that Dr. Sharon referenced, Dr. Cabral came down. It was unprecedented for us to have 3 cases in one summer. And I was working in epidemiology at that time in Orange County at the health department. And we just wondered, I mean, it had been 9 years since our previous case. And we were just unprepared for what happened that summer and said, oh my goodness, what's going on with Orlando? So she was gracious enough to come down and do a presentation for our staff and help us to ramp up, if you will, our awareness efforts. And so she actually came back later and did some samples and took us back to her lab that we were just so interested in what she had to share with us and the research she had been doing over the years that we were unaware of. And Jack Tracy, a colleague of mine, who was very instrumental in putting this together with me, he's the one who was able to connect with Dr. Cabral and get her here. And so we're very excited to have her join us again this year. And I'll say a few words about all of her experience here. She's a professor of microbiology and immunology, specializing in working with pathogenic free-living amoeba, including Naegleria fowleri and Acanthamoeba species, and Amoeba mandrillaris. And I'm not going to say these words correctly, probably. These amoeba are the causative agents of amoebic encephalitis. And of course, Naegleria fowleri causes a rapidly fatal disease of the central nervous system that we know now as primary amoebic meningoencephalitis. It's acquired from freshwater lakes and ponds, and as you heard this morning from other freshwater sources that we actually were not on the radar in years past. Acanthamoeba can cause amoebic keratitis in addition to encephalitis, and she'll talk more about that in her presentation. She studies the disease process and using in vitro methods to understand the mechanisms of invasion of the central nervous system. She received her PhD from the University of Connecticut, of course, in I won't say no, in 1974.
You can say it. You got to tell
She was
them
only 10.
Can you tell them when I was born?
I know, right? I didn't do that part. But it's my pleasure to introduce Dr. Marciano-Cabral.
Okay, yes. I've been working on amoebas for a long time, and I'm happy today when I saw the presentation of the child that survived. My heart was just taken up, and I'm thankful to God that I work on this organism, and I hope I have something to contribute. But anyway, I took this picture a long time ago, and somehow it got out, and that's how we got the name brain-eating amoeba. I don't ever call it that, but I guess there's a purpose for it. So I want to tell you about the studies we do in my laboratory. I'm a basic researcher, and we are trying to learn about the amoeba itself and what kinds of proteins it makes and just the basic chemistry and biology of this organism. So in my lab, our studies have focused on trying to identify virulence factors. These are components of the amoeba that can actually damage the host. And the purpose of our studies was we had hoped eventually if we could find virulence factors that were unique to the amoeba, then we might be able to find a target for therapeutic drugs and even develop a diagnostic test. So that was our intention, okay? These are just 3 of the virulence factors that we have studied in our laboratory. There are more. There are cytolytic proteins that we've studied that lyse the cells. But adhesins are important because these are surface proteins that allow the amoeba to attach to host cells. So when the amoeba goes in the nasal passages, it has these adhesins, and it allows the amoeba to firmly attach to the host cells. Those are integrin-like proteins. Then the amoeba synthesizes enzymes. There are some that I think are extremely important and are definitely virulence factors, and those are the proteases. They also make phospholipases. So these enzymes, my lab's been concentrating a lot on proteases now. And the other thing that's probably really, really important, all we know is that this amoeba has an ability to influence and induce a severe inflammatory response in the brain. All right, so the inflammatory response in the brain is really what causes a lot of the damage. These are called pro-inflammatory cytokines, like IL-1, IL-6, TNF, tumor necrosis factor. Those inflammatory cytokines are induced by the amoeba. So let me just show you some of the things, okay? We culture the amoeba exenically in our laboratory. What does that mean? It means we just grow the amoeba in media. There are no other cells there, and there are no bacteria. It's just in a growth media. Those amoeba then become attenuated. They have an attenuated state. That means they're weakly pathogenic. They're not as pathogenic as an amoeba that just comes out of an animal or a human. All right? Then an animal model was developed by others, and we use it in our lab to generate highly pathogenic Naegleria. I'm going to show you that mouse model. I hope you don't mind. The amoeba are mouse passaged at monthly intervals to maintain a highly pathogenic state. They're very virulent. And the highly pathogenic amoeba are compared to the weakly. All our studies, we compare highly virulent to weakly virulent or weakly pathogenic. Okay, so this is a brain from a girl that died in Virginia, where I live, in Richmond, Virginia. It was a long time ago in the 60s. We had a few cases then. And the amoeba was isolated from the cerebral spinal fluid of this 15-year-old female. And it was cultured in the lab, and then it was frozen away, and we took it out of the freezer and started growing it, and we've been working with it ever since. So we have the exenically grown, and now this is my little mouse model that I use in the lab, okay? You can instill amoebas in the nasal passages. You just put a few drops of amoeba in the nasal passages, and they will go to the brain, and once that happens, you start to see symptoms of the infection. The symptoms are the fur, the hair becomes, they call it ruffled, but it kind of stands up straight. The mouse keeps his head down. Sometimes they have seizures. They circle. They stop eating. So we have to sacrifice them now. That's the rule. You have to sacrifice them when they start showing symptoms of infection. And then we take the brain out, and well, I don't know if you can see it with the lights, but there's a hemorrhagic area there in the brain, okay? We then culture the brain to make sure that we can get the amoeba back, and that's what you'll see in the culture where we culture them again. So we can keep them like that for about 1 month or so in the media, growing them, and during that month we do all of our tests, and then we repassage the amoeba again so that we have a highly virulent strain. And it makes a big difference. So what do we see? Here's an uninfected brain, okay? And here's an infected brain. So in the uninfected brain, the olfactory bulb is fine. In the infected one, it's totally destroyed. And these are just H&E sections. You can make sections to look to find amoebas in there. But this is scanning electron microscopy that I do, and there's brain tissue here. It's all lysed. And that lysis is from some of the cytotoxic proteins and also from the proteases that are produced by the amoeba. Okay, so there's the amoeba sitting there with its food. We call that a food cup, and that's what it uses to ingest the brain tissue or bacteria out in the environment. So how do we know that our mouse passaged are highly virulent? Well, we've done what we call infectious dose 50 test. So we can use the same number of amoebae, okay, in a mouse passage one, or an exenic one. And if you just look at these, it takes 2 times 10 to the 3 to kill 50% of the mice, or I can't say kill, they're infected, okay, infectious dose. Whereas still with the exenic, none of them. We get nothing, okay? So there's a huge difference between a mouse passage. And I think, you know, when you do drug studies, it's going to be really important to try some of these highly virulent ones because they may be the ones that'll take the drug in. And, you know, if the drug sticks to the surface of the amoeba, these amoeba are highly phagocytic. That's the problem with antibodies. When you that's why a lot of antibodies are not effective against the amoeba, because an antibody that attaches to surface proteins, they'll ingest them and destroy them, and then the amoeba just goes walking away. And so there's a lot of things we have to consider, I think, okay? So our purpose of our studies is to try to, determine differences between weakly pathogenic and highly pathogenic amoeba. We want to see what the virulence factors, if they're totally increased, and what those virulence factors are. We hope to define some of those maybe as a definition for a molecular target for therapeutic intervention. And maybe some of those will be for a rapid test. I don't know, diagnostic test. We haven't gotten that far yet. So what do? We're looking for expression of proteins by Western immunoblot. We prepare what we call host cell lysates of the weakly pathogenic and the highly pathogenic, and then we compare their protein expression. We even collect media, okay, so that we can get the secreted proteins that the amoeba are putting out into media and secreting. And then we prepare the membrane preps of the amoeba so we can get these proteases. Okay, then what happens? All right, the first thing we looked at were these, the integrins. Now, you know, it's really going to be difficult for drug discovery, I believe, only because, I'll tell you why, there's a lot of proteins that have similarities to human proteins. And so you got to make sure that when you find something, you are directing it to a unique protein in the amoeba and not a human protein. And one of the problems, okay, so this is a virulence factor, this beta-1 integrin, because it's all over the surface. This is an amoeba, these are immunofluorescents, and there's phalloidin, okay? These, I don't know if you can see it very well, but this is all one big amoeba here, and these are all integrins on the surface. Okay. What happens is, though, that we use an antibody, a human antibody, an antibody to the human beta-1 integrin to detect it by Western blot analysis, right? So that means that integrin in the amoeba is very similar to the human protein. That would not be a good target, all right? Even though it is considered a virulence factor. I think a really important virulence factor is whatever is causing these amoeba to induce pro-inflammatory cytokines. And this is just Naegleria fowleri, that's LPS from bacteria, in a comparison with mixed glial cells, which are brain cells, primary brain cells that we get from rats. And if you look at them, Naegleria fowleri and LPS from bacteria are inducing about equivalent amounts almost of these pro-inflammatory cytokines. Additionally, when we compared the mouse passage to the exenically grown, the mouse passage induced a greater amount of pro-inflammatory cytokines. So it's more virulent in that sense. Okay, what else have we done? Okay, we have been concentrating now on proteases, enzymes that break down proteins. And so this is just different concentrations of the amoeba, but this is what we call a host cell lysate. We grow up the amoeba and then we wash them we spin them down, we get a pellet, and we freeze thaw the amoeba a couple times, and then we use that as our protein. Okay, this is called zymography. It is electrophoretic separation of proteins. But in this gel, it's a gelatin gel, all right? It's because it's gelatin, whenever you see these bands like this, it means that the amoeba have produced or have proteases that break gelatin or recognize gelatin, all right? So if you just to look at axenic and mouse passage, what you see here is that the mouse passage is making more proteases, a greater amount. And we've even noticed that there's some that are produced in the mouse passage that we really don't even see in the exenic. So I think proteases are very important. They're used, definitely we've shown they're used in invasion. We have an invasion assay in our laboratory that we use. This is a well here, and this is the plate we put the well in. In the well, we use something called Matrigel. Matrigel is a commercially available extracellular matrix material. We put the amoebas on top of this, okay? There's Matrigel. This is the well. We put the amoebas here, and they have to get through that Matrigel and get to the bottom. That's the invasion. So this is the top, and this is the bottom. Here's the amoeba. Now they have to secrete all these proteases to invade. And that's what you see here. This is the top of the well here, and we've got proteases being secreted, okay? There's a few in the bottom, but it's really the top that's important because that's where the amoeba are secreting the proteases to get through that Matrigel. So I think proteases are important. And they're important virulence factors for a number of reasons. These are the cells we grow. These are mixed glial cell cultures, astrocytes, and microglia, which are the immune cells in the brain. What happens is, it's not only that the amoeba are making proteases, but these cells also are secreting proteases. Right? So if you look at the live amoeba, yeah, it's lysing cells everywhere. But if you collect the conditioned media from the amoeba, which means they're secreting their proteases, they are also lysing the cells in the brain, okay? And so the proteases, I think, are important because one of the things they do, and I just learned this from, reading the literature, studies by Rosenberg et al., there's a paper in Glia, he's got several papers. He talks about proteases in the central nervous system. And these proteases play a critical role in CNS inflammation. He's studying them in terms of a number of neurological diseases like Alzheimer's and a whole bunch of different neurological diseases. And he finds that the proteases are playing the critical role. And it's the neuroinflammation that's causing all these problems in these neurological disorders, okay? So the point I think I'm trying to make is that proteases are in the CNS. They can cause disruption of the blood-brain barrier. That leads to cerebral edema and hemorrhage. And you sort of got a double whammy now when you have an amoebic infection, because the amoeba are making proteases and the cells in the brain themselves are making proteases, okay? So you're getting tremendous neural inflammation. And he suggests that protease inhibitors be used in some of these diseases. And he states that protease inhibitors have been used to treat animal models of protozoan and neurological diseases. Plasmodium, Leishmania, and toxo, and even trypanosomes. They've used protease inhibitors. These were in animal models, however, okay? So I think you have to, take that and realize that maybe they work in animal models, but are they going to work in human models? Humans, we'll have to find out. Okay, so that's about, that's about it. I just want to have a special thanks to Jack Tracy and Leanne DeGrandis, to the Florida Hospital for Children, and to the families of the PAM victims, Jordan Smelski, Kyle Lewis, Philip Goff, who have helped make others aware of the disease to prevent further infection. I think we owe a ton of gratitude to all these people. And I'd like to thank Donna also, because I just met you this year, so I'm happy to meet you. Thank you. Let me just show you this. This is a paper, I found it in a drawer at school, at my school, okay? And it's the Orlando Sentinel from can't even see it, but whatever. Yeah? Okay. Beware, lethal amoeba lurks. And that's that bad word. That's our rare, but that's the bad word. We don't say that anymore, okay? And this teenager caught it in a canal. And then Jack Tracy had invited me to come down and go collect water samples. That's when I came. Look at that. 9 out of 10 water samples from Florida lakes were positive by PCR. We take the water back to our lab. I put it in a petri dish. We can put it on agar with bacteria. And if there's amoebas in there, they'll eat all the bacteria and they'll clear. You'll find these clear spots. And then we collected the amoeba and we did PCR on them. And sure enough, 9 out of 10 were positive. So I think we have to really keep in mind that this is an area where it's prevalent in the water, okay? And I just want to thank you for your attention. And if you have any questions, I'm more than happy. I was just going to say one thing. I wanted to make a comment this morning when they were talking about recognizing the amoeba in culture. When they culture, the amoeba, Naegleria, has a very unique nucleus, okay? It has a very large nucleolus. And if you look at the amoeba, you'll see like a black area, circle area inside the nucleus. That's always a tip-off that you've got an amoeba versus like a lymphocyte or some other cell. So that's a good, I wished I had a picture of it, but I don't.
Question. In the case, are you familiar with the case in North Carolina, the Whitewater case? I was just curious how that, because that's an artificial, but I know that they were trying to make it natural for the rafts and the trains. So how is it that got so much of the amoeba there and they didn't even know it?
Is there, I don't know that area, but do you know if there was like a lot of dirt around?
I'm from that area. I'm from
There's a lot
Charlotte.
of dirt there?
They dug all that out and made it artificial. It used to be trees, lawns. It's near the airport.
Yeah.
So it is a lot of dirt, but they have a lot of rocks they took from the lakes around Charlotte to make that artificial. But I was just curious.
I know, it's hard to tell you, if you notice, if you go in the swimming pool, if the swimming pool has cement around it, that's a lot better than if it has grass and dirt, because the amoeba can live in moist soil.
Well, there's cement around it, but they have the natural rocks they took. I don't know.
So the problem with North Carolina was a few different aspects where their UV treatment wasn't at good standard, and the organic material within the water was obviously having a play on how well the water system was getting treated. So the water was constantly getting recycled as well. So if you're recycling untreated water, obviously the organic material is going to increase, so the susceptibility to the UV treatment is going to decrease over time.
So they just didn't have education about recycling the water and treatment properly?
Yeah.
Well, because that's been there for like 5 years.
Yeah, it's been there for
It's been
a
a
while.
while.
It was
Thank you for helping.
the CDC that pointed out
Yeah.
that there was a few sort of
They
recommendations
stopped
that-
the whitewater center
Yeah, I
for
know.
several weeks, yeah, and then they
It's back
treated.
open.
Yeah.
I didn't know that area. Oh, thank you, thank you. So,
Okay, my question about the water samples, I'm from Central Florida, and under what, what different lakes were tested? Were they tested in the middle, were they tested on the edge? Those kind of,
well, there's always this
I was just curious.
thought that they should be tested around the edge
That's
because,
where people swim.
yeah, and, they're on the bottom. But I think people forget that these amoeba can flagellate and swim to the surface.
Right.
And that is exactly what they do. They can swim to the surface because they eat bacteria. So they swim up and they actually, I don't know how they do it, but they can, turn back into an amoeba again at the surface, get the bacteria, and then they'll swim back and go down. But the bad thing is to disrupt the bottom, really. That's, they're mostly there around the edges and in the shallow water because they're in the bottom. But they can be at the surface, so you never know. And I had this other, wait, and you know what? I think I had one more slide. Oh, I don't know what happened to it. That's okay. It was, why do some people get Naegleria and how come everybody that's exposed doesn't get it? I have a couple of theories, okay? One of them is, and it's been shown by some scientists, that if you have an IgA deficiency in the nasal passages, it could be a reason. I think that maybe some kids or people get infected with a very virulent strain. I know that in addition to the amoeba that we work with in my laboratory, I have a second strain that I got from the CDC. And we were comparing these two totally different Naegleria fowleri strains. Not even thinking about exenic versus mouse passage. That's what we're at now. And two different strains from two different isolates from two different humans. And there's a difference in them. The one that I work with in my lab is really pathogenic. The other strain that I got, it's pathogenic, but it takes a lot more amoeba to cause an infection. So maybe some children have got infected with these different strains that are extremely virulent. And the second point, I think, is we know from our mouse studies that when the amoeba come out of the mouse, they're more virulent. We know that there have been animals in nature that have been infected. Maybe the kids got infected with an amoeba that just came out of an animal. I don't know.
I have one other quick question. So what, and he probably didn't do this, but I'm just curious, what concentration were the amoeba found in those lakes versus the concentration of bacteria, because amoeba eat bacteria? I'm just trying to compare small
Well, we
organisms to
didn't-
one another to see.
Unfortunately, we didn't really do that.
Okay,
We
that was
just,
just
I'm
curious.
sorry.
That's
Yeah,
okay, I'm
maybe
just
somebody
curious.
else knows. You
okay.
know, go for it.
That's actually my project 30 years ago.
Oh, really?
I did that, and it falls on what Francine said. First of all, you don't just look around the edges.
Right.
We found it actually, the surface film, that little layer on the very top, we took little loops and we would find the Naegleria there. We found Naegleria in the thermocline where the water temperature changes. And all it is an amoeba is just another small particle that float down and they slow their descent on that temperature change. And you find lots of bacteria, algae, and amoeba there. So to go back to your question, we were sampling all these different areas in these lakes in upper part of South Carolina. And when we started finding Naegleria fowleri, we would find them anywhere from 1 every 50 mLs of water to sometimes 20 per 10 mLs of water. So the concentration can range quite, and even within a water column from this tall, from my height to the bottom of the thermocline, we would see some more amoeba here, less, and then we would see a lot at the thermocline. But if the wind came in, that all the water on the top is about the same temperature, it starts stirring up, so then all the amoeba are stirred up. And that's without people swimming, without cows moving the sediment, whatever. So there's a lot of different ways. So I think Francine's question, what causes some people to get infected and others, we really don't know if it's numbers, pathogenicity, but those are things we could test for.
Thank you.
Yeah. I think, yeah.
Is anyone planning to research the prevalence or frequency of highly virulent amoebas versus not so virulent bacteria? Amoebas found in lakes?
From lakes,
Yeah.
that's a good thing to do. I could do that because I have an animal protocol that's been approved where I can just take amoebas and infect mice with them.
Yeah.
That's a really good idea, just go and find out and see what's going on.
Because you could have like differences in the prevalence of highly virulent amoebas in different geographic
Yeah.
regions.
That's really a good idea. Fantastic. I mean, nobody's planning on doing that I know of, but you know, that's a good idea. Do you want to do it?
Sure, I'm clinical.
We're going to convert you to research.
I'm clinical, but
It's
I'd
okay,
love
we
to.
need all kinds of people.
Now, the other question is, I've been involved in the care of some of these patients, including our last patient here. I personally had a question whether, because everyone talks about how amoeba eats the brain.
Yeah.
I haven't found any data on what about the rest of the central nervous system. Because, so the amoeba is obtained from a sample of CSF that is found, that surrounds the entire central nervous system, right? So what about the spine?
I think, well, it would be my, I don't know, and let me just say that from our animal studies, when we were allowed to let the mice die, we just can't do that anymore, but when we did, the amoeba did go back more into the brain, but then depending on how many amoeba you give the mouse, that's really important, the number of amoeba. And we could find them in the lungs, as a matter of fact.
Well, did
Okay.
Philip Gump get it in his heart?
In Philip Gump's case, it was in his lungs and it was in his heart.
I'm sorry.
Philip Gump?
Philip Gump,
Yeah.
Sandra and Tim's son?
Yeah.
It was in his lungs and his heart.
Yeah, I know, because if you get so many, it goes up the nose, but then what happens is it goes down into the lungs too. It's terrible.
Yeah, and the reason I'm asking that is because as we probably move into a situation where we start having more patients survive this illness, we could start seeing some residual effects that haven't been detected before. Before most patients have died. And so the main outcome that we see is, of course, severe brain injuries. But if there's more subtle injuries that will still lead to neurological deficits, I'd be interested in knowing
I'm
that.
sure you're
Like,
right,
your
yeah.
animal models are there. Is there involvement of the rest of the CNS?
That's, we'll have to look, I'll look. I mean,
Uh,
you looked?
Years ago, a guy named A.J. Martinez from Pittsburgh did some studies, and his conclusions were that Naegleria tended to affect the frontal part of the brain because that's where they came in.
Mhm.
And so they start to do their damage. Whereas with Amoeba, and if he can show you, those are spread by hematogenous spread. So they come through the bloodstream, and so you can find those in all parts of the brain. So I think a lot of it is just, that is the access point where Naegleria gets in, and that's where we see most of the damage. But with the pathology, of course, as you know, these can be spread back into the blood, to the heart, to the lungs, to other places. Steve?
So I had a question.
Should we be giving the microphone to people, or
So you can pick it up?
It would definitely help, yeah.
Okay.
Thank you. All right, so,
It only goes to the camera.
okay. I have a question. Now, you mentioned how it actually attaches in the nasal passageway. How many attach, and is there any type of rinse or cleansing or anything that you could do after swimming that could affect whether it actually gets in or not?
Start squirting salt water up there right away. That's, I mean, just, you know, nasal spray. Make sure it's saline though. And I mean, the people that have done the neti pots, if they had boiled the water, it would never have happened. All you have to do is boil your water. And we had a couple cases in Arizona a few years ago where kids in the same neighborhood died. They had the same water company supplied water to them, and that water company was a private water company that did not chlorinate the water. So 2 kids in the same neighborhood died from that. But I just say,
what is the salt content that actually would kill it? What's the salt level? So in Florida, we have chlorine pools and we have salt pools.
I know.
And we keep it between 3,000 and 3,300 parts per million for salt.
What do you
Is that
think?
enough to kill it?
Do you know?
I can't remember those data.
I know.
I can go back and look. I think that was done a long time ago.
Yeah.
Do you know?
Jennifer will know it.
Jennifer might know that, okay.
Yeah, cool.
Okay.
She will know.
Because that was something that we thought about doing, is seeing if Florida would pass a law making all new pools going forward salt pools rather than chlorinated pools.
Yeah.
Because you can let your chlorine down, but if there's salt and if there's a level that kills it, maybe
Yeah.
it
And
would be
there's
So
safer.
one problem I can foresee with that is we've actually identified highly osmotolerant Amoeba in the environment.
Okay.
So if, obviously, it can be similar to Naegleria, we don't know.
Okay.
Like, we don't know how much salt is actually needed to that extent, and can we build a resistance from the concentration of the salt. And then it comes back down to if it's a non-properly treated pool, like chlorine, are you going to see these sort of resistance, sort of Naegleria as well, you know? So I think maybe the combination of both would in fact help in that aspect.
Do you know about chlorine? There's something else, chlorine. Do you know
Yeah.
about chlorine?
Well,
Is it
maybe.
better than chlorine?
I think it burns off faster though, but I don't
Oh,
know.
that's right, yeah, okay. Never mind, forget it.
We have thousands of questions, but should we let Ibne say a few words? Go ahead. We need you to say a few words.
Can I chime in?
Yeah.
We're going to go to lunch at noon, and so we've got about 6 minutes, and then they want us to go in for a prayer before we go.
So we've never even gotten to all our questions yet.
We need to pick a few, so you can, after lunch, talk about some areas that you'd like to focus on.
So do you?
I'm a woman.
So we were asked to pose some of these questions about research areas that are going on currently, nationally and internationally. I think you heard there's a little bit of drug discovery work, there's a little bit of diagnostic work, but there are very few groups working on that. And sometimes people that don't do science don't appreciate how important the basic work is, and very little of that's going on to understand new targets. So which of these would be most directly applicable to prevention of PAM deaths? We're hoping that drug discovery would be. We also hope that rapid diagnostics would be. Is there anything else that you would add to the list of what is the most important that would prevent PAM deaths?
Yeah, what thing, besides this thing, other thing is the awareness, increase the awareness
Right.
among people and clinicians.
These are for research agenda.
Oh, yeah, and training for research. I think we need to find out definitely better drug and better diagnostic, and also we need to find out if there are differences in virulence. We need to have a better genotyping tools to categorize them into more number of genotypes, so we can check like whether only certain genotypes are more occurring in the PEM patient. So we need a better genotyping tools.
I think his suggestion was really good. Do we know about where you find them in the environment?
Yeah.
Are they really highly in the environment versus weakly?
That's right, yeah.
You know,
Yeah.
that kind of thing.
That's right, yeah. If we have a very good genotyping tool, then we should be able to answer that question. But currently, our genotyping system only categorizes them into 3 groups, and they do not show any link to anything. Yeah, so we need a better tool. Yeah.
So one of the other questions were, do virtual research communities now exist? I'm not aware of anything for this disease. There are some sort of open source drug discovery for malaria and other diseases that more people work on, but I'm not aware of anything for this work.
No, I don't know.
And I guess the question would be, is there interest, and even in our group, for there to be a virtual community for that?
I don't see, I think it's just accumulating information and having people interact is always a good thing. So
Yeah.
I don't think that's a problem. So establishing one, yeah. So there's a lot of interest in this core question, and that is, why do some people get infected and others don't? I think what my interpretation is, there's a lot of different potential explanations. Francine gave a few. But really what we need is some new tools. And what Ibne talked about this morning with the whole genome sequencing, that's going to open up a door so that we can really understand what is the variability in the ones that are pathogenic. And if we can understand that at a basic level, then we can ask good scientific questions about what is it that makes this pathogenic. And then we can also maybe test models. We could use animal models to test ideas for why does some adhere to the epithelium, why does some go through and get into the brain. Those are the kind of studies that nobody's really done well, in my view. And I think there's one other question. We keep hearing this. People keep saying that it's the trophozoite. I've never seen really good data that says you can't be infected with a cyst. I've never seen really good data that a flagellate can infect somebody. And we just, maybe it's just that in warm water there's a lot more amoeba there, but in cold water they're just as potentially deadly if you get it up your nose. People just aren't swimming. So there's some really basic things that could be done. So I think one of the things they want us to do is, what are the key questions that we want to propose? If we were going to try to have a research agenda where we could get some funding to do work, what would be the key things to be done? So I'd really love to hear from the people who are not the ones working on it. What do you think?
Yes.
Yes to all?
Yeah, well, understanding better about how it actually reacts within the body.
The early prevention, I remember last year, not early prevention, early detection. I remember last year you were talking about a possible nasal swab. So you're catching it without doing all the intrusive things that have to be done, and possibly even catching it sooner than a lab technician that maybe isn't trained in it. I think those are two things that come to mind.
Are we testing all the freshwater lakes around Central Florida to kind of know, like, okay, May maybe have more, July has a little bit more, August, and kind of keeping signs around those areas saying, hey, this area has this amoeba in it, do not swim during this time. When the film, is there any kind of research or testing environmental health going on or possible?
It's really big in Australia, though. Their water monitoring system. It's definitely something that could be adopted in Florida.
Yeah.
Well, Beatrice is saying there's groups in Australia and in France that they have labs that are devoted to testing for these pathogens in water. Because Francine, we heard about this morning, piping water over the arid parts of Australia. That's how they came up with a lot of the early cases, and so they have a detection system that's still in place, and they have, what is it, Chris? How many amoeba per liter? That they have a detection limit if they find 10 to 100. Yeah, so it's very, very low, whereas in France they have a different detection limit. Why? We don't even understand that. There's nothing like that, although in Louisiana there was a case, right? Do you know anything about?
Yeah.
About the water testing?
Yeah, they found Naegleria in the main water line, drinking water line. And when they treated it, then it's gone. But they did find it, except
they
I
had some
don't.
ongoing testing too, and then those people actually were putting false data out.
Oh, I don't know this.
Yeah, they weren't doing the testing.
Oh, I think I heard that.
Yeah.
That they were not even testing it. They are just giving false data that it's negative. But eventually found out that they were not very truthful. Yeah, that's right.
One of the challenges with signage, and that's something that came up, in our 2007 cases, is that if you post a sign in one area, then people think it's not in another freshwater body, but it's naturally occurring. So you can't say that it's not in a lake or a pond or a canal. And so that presented some challenges. And so, what Orange County did at that time is they posted it on the bathing areas, so all those lakes that were considered bathing areas, they went ahead and posted along with the county, so the health department in the county. But they also put up other messages. So watch out for the alligators and don't dive headfirst. And so they used that opportunity to list out about 10 different things that you should take precautions with when you're participating in recreational water activities.
Has it even been considered starting elementary school all the way?
Has it been even considered in elementary school, even in health class, to teach the kids about the amoeba, teach them about swimming, teach them about what not to do, what to do when going to fresh lakes in Central Florida and other states? Has that even been a consideration?
Yes.
The education, because the kids will teach the adults.
Yeah, I can talk to that a little bit too.
I mean, that's part of the education campaign that we do every year, is we put the information out in backpacks before spring break. We put it out in backpacks and online. It's, of course, electronic and social media and reverse calls. The Seminole County Public Schools does that, does a reverse call before summer vacation, those months where it's more prevalent. But those campaigns are there, they're not enough. But those campaigns, are there. We just have to continue them.
One more, and then we have to go.
I think you were asking for where research should be headed. I think looking into some of the basic substances that kill amoebas that we could potentially use intranasally, as a preventive measure. Like every time someone swims in a lake, why can't they just spray something up their nose, like alcohol-based or whatever it is? I don't know what kills the amoeba, but something that is tolerable by the nasal mucosa. Is that something that we could do for everyone and just see how the rate of PAM declines? Yeah, because something alcohol-based could potentially be tolerated by the nasal mucosa, I think.
I think now that's
Steve, do you want to?
No, I think you guys will just have to come up with your list. We're going to go back and have a prayer before we eat. And
Oh, go back
you guys
in.
Go up on stage and actually present on which areas that we're going to go after.
I wasn't present, so I got selected. So, yeah, we'll do something. Don't worry. And if anybody has any comments or questions about what we said, we're all here. We'd love to hear from you. Talk to anybody.
You'll have opportunity